Hello, SoilCon. My name is Kwenama Buthelezi. I'm a PhD candidate at the University of KwaZulu Natal in South Africa. Today, I'm excited to share my research proposal, which is a project generously funded by Potato South Africa. We are exploring a multi-scale approach to precision agriculture, specifically how we can integrate sentinel satellite data with high-resolution drone imagery. Our goal is to accurately monitor the soil parameters and model long-term soil health within potato maize rotation systems here in South Africa. Because this research is in its early stages, I am very much looking forward to your feedback and insights at the end of this session or during the questions. To guide this research, we are addressing four critical questions. First, which soil parameters exhibit the highest spatial variability in South African potato cultivation, and which of these are most vital to crop success? Second, one to identify those parameters, which specific spectral bands and vegetation indices from our satellites and drone data correlate more strongly with them. And third, which machine learning algorithm will allow us to produce the most accurate and reliable soil health prediction. And finally, can these predictive outputs be successfully transferred into actionable management zones that farmers can use on the ground? Ultimately, we aren't just trying to look for data. We are looking for a workflow that moves from a complex imagery to practical field management practices. So to answer these questions, our project is built around four primary objectives. First, we aim to develop a comprehensive integrated monitoring and prediction framework specifically tailored for potato maize rotation system. Second, we will try to quantify the spatial variability of key soil health indicators across our various sampled farms, establishing a robust baseline for our data. Third, we will extract and analyze full season satellite indices, relating these spectral signatures directly to soil health and potato development stages. Finally, the culmination of this work is to integrate our model outputs into practical decision support framework, of which will provide Potato South Africa and its farmers with clear science-based tools for managing soil health in real time. We will be taking soil samples from potato field selected in collaboration with Potato South Africa across different regions and climates. The soil samples will be taken using grid sampling on three stages, which are pre-planting, mid-season, and post-harvest, to establish the ground truth data for physical and chemical soil health indicators. Then we will extract satellite and drone images and generate predictive algorithms of soil health maps, management zones, and fertilizer or irrigation recommendations, which are tailored around potato production. So in closing, in this collaboration with Potato South Africa is a vital part of our broader innovation and transformation goals. By providing locally calibrated soil health diagnostics, we are moving away from generic model and toward a solution specifically designed for South African potato maize rotation. Beyond immediate productivity, this project serves a larger sustainability mission, developing a robust monitoring tool to track and mitigate soil degradation. So our aim is to ensure that our agricultural system remains resilient and productive for generations to come. Thank you for your time, and I look forward to your questions and feedback. Thank you.
Hi everyone, I'm Isha, a master's student at Washington State University and working currently with Dr. Gabriel Lehu. Today I'm going to talk about how soil compaction affect potato yield and soil moisture. To give you some background, in the maritime Pacific Northwest, the interannual variation in precipitation and competing demand for water resources is imposing a serious challenge for growers. On top of that, the intensive and repeated field activities induce surface and subsurface compaction, which restricts the root growth, affect the water holding capacity, and limits the availability of water to plants. So, to address these challenges, we are investigating how water application rates, soil moisture threshold at irrigation, soil compaction, and their interaction affect potato yield and soil moisture availability. For today's talk, I'll be focusing on soil compaction. This slide is here to give you an idea about how our research trial looks like. The main plot is the irrigation treatment where we have three different level of water applied per irrigation event and three different soil moisture threshold at irrigation. The main plot, the irrigation treatment, is further splitted into compacted and uncompacted plots. The compaction treatment here, what I mean is the rototilling was done shortly after the rainfall, which was followed by field traffic in compacted plots, whereas in uncompacted plot we waited for a few days and then do the rototilling when the moisture was lower. Along with this, we do the chisel plow just prior to the final pass of rototill. For assessing the soil compaction, we measure the pentometer resistance. In the graph, you can see on x-axis, we have pentometer resistance in megapascals, and in y-axis, the vertical axis, you can see soil depth in centimeter. Here, the dotted line represent the pentometer resistance value for compacted plots. At the depth of 20 centimeter in 2024 and at the depth of 17.5 centimeter in year 2025, you can see a sharp increase in pentometer resistance value for compacted plot as compared to uncompacted plots. As another measure for soil compaction, we assessed ball density. For this, we took the soil sample to the soil depth of 60 centimeter, then divided the whole soil column into equal segments of 7.5 centimeter and assessed the ball density. By this, we found that there was a significant difference in ball density between the compacted and uncompacted plots at two soil depth, and they were 22.5 centimeter to 30 centimeter and 30 to 37.5 centimeter. Moving towards the result related to yield, here in the graph you can see on x-axis I have the compaction treatment and y-axis I have potato yield, which is in metric ton per hectare. And in year 2024, we do find a difference between the compacted and uncompacted plots in terms of yield, and soil compaction reduced the yield by 7% as compared to the uncompacted plots. But in year 2025, there was no significant effect. Finally, to understand the effect of soil compaction on soil moisture, we use the data related to soil matrix potential. So basically, we plotted a graph of soil matrix potential across the growing season. And interestingly, in year 2024, we found that in one of the 10 irrigation treatment, the compaction limited the amount of water plant could access as compared to uncompacted conditions. And in year 2025, we do find similar results in 3 out of the 10 irrigation treatment. So definitely we need further research to investigate to identify the condition where soil compaction affect the yield and soil moisture most. And lastly, we would like to acknowledge the support provided by Washington State Department of Agriculture through Specialty Crop Block Grant Program. And we also like to express our thanks to all our collaborators. Thank you so much.
My name is Paul Martinez, and I'm a graduate student at Washington State University working with Dr. Gabriel Hugh. Today I'll be presenting on how does organic management influence soil susceptibility to compaction, plant below water, and water movement. A brief introduction on soil organic matter. It is well accepted that soil organic matter helps increase what is operation defined as plant available water and also increases saturated hydraulic conductivity. Similarly, soil organic matter helps decrease soil susceptibility to compaction by improving resistance and recovery. However, it is unclear if the effect of soil organic matter has on soil susceptibility to compaction can magnify its effect on hydraulic functions, especially on soils that are prone to compaction. To carry this out, we took soil samples from Russell Ranch Center experiment at UC Davis. There they had a 2-year crop rotation with tomatoes and corn, and we took samples from the organic and the conventional systems that they had in the experiment. The organic management consisted of composted chicken manure and winter cover crops, and then the conventional management system used synthetic fertilizers and left their fields winter fallow. We took 48 samples at a depth of 15 centimeters, and each sample was 5 centimeters in height. When we brought the samples back to the lab, we adjusted the moisture content to 80% saturation, and then we compressed them at 200 kilopascals, and this is only half of the samples. And for those samples, we took measurements right after the compression treatment. For all the samples, we took lab saturated hydraulic conductivity, and then we also developed soil moisture release curves with using the HyProp2 and then WP4C. And then the best fitted model for our samples was the bimodal PDI Kusoji model. And then based on the results, we observed that management did not have an effect on soil susceptibility to compaction or recovery. And we measured that through soil resistance, which was the difference in bulk density of the compacted and the uncompacted samples. And then soil rebound was the difference between height reduction right after compaction and then 24 hours after compaction. However, although we did not see an impact from compaction on recovery and rebound, we did see an impact on KSAT, and we observed that there was a reduction in KSAT from compaction. However, there was no difference in KSAT between organic and conventional systems. We also developed soil moisture release curves, and with this information, we could calculate what is considered what is operation defined as plant available water, which is the difference in volumetric water content at field capacity and permanent wilting point. And then on the right side, I have this table summarizing the trends that we observe on the data from the soil moisture release curves. So with the organic management, we observe an increasing trend at saturation, at field capacity, and plant available water. And then we also, with the compaction treatment, we observe an increasing trend at field capacity, at permanent wilting point, and plant available water. And I just want to reiterate that plant available water is operationally defined, um, such that compaction would not mean that water availability increases for plants. Um, Plant available water is only considering what's between field capacity and permanent wilting point, and is not considering anything before that, or any effects or any structural impacts that compaction has on soil. And these are my takeaways. 25 years of organic management, we did not observe a reduction in soil susceptibility to compaction. However, compaction did reduce water movement through soil. And then lastly, that plant available water increased with organic management. Thank you.
Hi everyone, my name is Leslie Michael and I am a soil scientist at WSDA. One of the projects that I get to work on is the WashE Soil Health Ambassador Program. So today I'm going to share a little bit about the program and highlight a few of our producers from 2025. So the Soil Health Ambassador Program was created to highlight the innovative soil health practices that are adopted across the state. So far, we've highlighted a variety of different projects such as cover crops, making and applying compost, and utilizing livestock to improve soils. Our goal is not just to share the incredible work that farmers are doing across the state, but also to connect farmers to each other. So if you're interested in one of the practices a farmer implements, if you have questions, or you just want to connect with a fellow soil health enthusiast, definitely reach out and we will get you connected to them. Our ambassadors are so passionate about soil health, they are happy to share their experiences with others. We're always looking for farmers or ranchers across the state that are using new soil health practices or techniques and might be interested in participating. Producers receive a stipend for their time, so if you or someone you know might be interested, definitely reach out. So let's talk about a few of our ambassadors this year. I provided QR codes so those will link you directly to the blog post and their video. I'll let them do most of the talking, but I did want to share a few things about each of them. This spring, we visited with Doug Schuster to talk about applying compost in dryland wheat. I was really inspired by Doug's just very practical approach to soil health. He's navigated a lot of different challenges, such as equipment, budgets, and the environmental impacts that come with applying compost with heavy machinery on steep slopes. Next, we have Reed Wilson. He's the farm manager for Hedges Family Estate. He shares about removing burns in vineyards to improve soil health. Reed has worked in so many different places on the West Coast, and so he's just has a wealth of experience and knowledge both about soil health and viticulture. I'm really excited to see what other innovative technologies he adopts over the coming years. And finally, we talked to Scott Chichester. He's the owner of Cheese Farm, and he has been mowing cover crops and just figuring out that system. Scott is so incredibly passionate about soil health and also growing nutritious and delicious, I've tried it, produce for his community. He's found a system that really works for his production and is worth hearing from. I really continue to be inspired by the farmers that I meet and talk to. These videos could be so much longer than 5 minutes, kind of like this one. But again, I encourage you to reach out. You can reach out directly to me or through our website. There are links both for nominating someone who you think might be a good soil health ambassador, but also if you want to ask a question, I'll connect you with our ambassadors. Also, be sure to sign up for our newsletter. The next video that's coming up, we'll be talking about using drone technology to reduce compaction. Thank you.
Hi everyone, it's a pleasure to have you listen to my talk. My name is Benedict Okorie. I'm a PhD student at Utah State University, and I'm working under Professor Martinez. And today I'll be talking about one of my PhD projects, and it's on soil microbial community responses to biostimulant application in Utah farms. And we use the phospholipid fatty acid profile analysis for this study. Now, I would like to give us a little bit of background to this study. Utah is one of the driest states in the US, actually the third driest state. So this means high heat accumulation in the soil and everything underneath the soil, including the microbes and the plant roots. Droughts are also common and they are unpredictable and most of the times. And then we have salinity issues. And that's because much of Utah's valley is underlain by ancient salt-rich sediment, which predisposes the soils to high salinity levels. So all of these factors together build up a high stress condition for soil microbes, as well as the crops that are grown in the soil. So the objective of this study was to evaluate the effect of biostimulant applications on soil microbial communities, their activities, and their dynamics. So we chose phospholipid fatty acid analysis because it offers a rapid and sensitive assessment of living soil microbial communities, total microbial biomass, and community compositions. So we had this study set out in 3 sites in Utah, the Squaw Valley, Nephi, and Delta sites. So we laid this out in a randomized, completely block design with 4 replications in the fall of 2025. And the biostimulant product that we applied was Regenigro product, which is a commercial biostimulant derived from microalgae and biochar. So we apply this 1 to 3 times in each of the sites via foliar spraying at the rate of 9.4 liters per hectare. That's about 1 gallon per acre. So at the end of the growing season, we went back to take soil samples from those sites, and we sent those soil samples to Lab for PLFA analysis. And we got the results back and submitted the results to data analysis using R. So the results, so far, it showed both successes and failures, and that's one of the things that has continually characterized field assessment of microbial products. So out of the 3 sites, we found significant differences in some of the parameters in only 1 site. So, but the preliminary results that we saw, which you can see here, that indicate measurable shifts in microbial biomass following biostimulant application. So we could see higher functional group diversity index. We could see higher total bacteria, higher fungi, higher saprophytes biomass, as well as higher gram-positive and gram-negative biomass. And that's what's interesting in this study. The only 1 instance where we had a higher amount of the biomass was for the undifferentiated biomass, in which case we had a higher value for the control relative to the biostimulant plots. But that was just 1 case in all of the significant ones that we saw. So it was interesting that the plots that received this product had a higher ratio of precursor to cyclopropyl, and this is an indication of active microbial growth, even though the value that we saw here, it's not that high, but then we saw that it was higher in the plots that received the product compared to the control. And that's a kind of promising. So I want to point out that this site, the Squaw Valley site, was characterized by high salinity and drought stress conditions, unlike those other 2 sites. In fact, the Delta site was almost always well watered, but that was not the case in this site. So, and there are evidence in the literature to suggest that under stress conditions, that microbial inoculations, they tend to perform better compared to a relatively favorable condition. And that could be one of the explanations that we could use for the explanation of this results that we got. So, but we're hoping to repeat this study this year and to further validate these findings. Yeah, so giving a shout out to the partners in this grant, Utah Department of Ag and Utah Soil Health Partnership. And thank you guys, and I'm more than happy to take questions as they come.
Hi, my name is Adam Peterson, and I'm the WSDA agronomist. My lightning talk today is about how the right tools can make nutrient management easier and more accessible. So we've heard a lot about the 4 R's, right rate, right place, right time, and right source. But I'd argue there's a 5th R, and that's having the right tools. Nutrient management is complex by its nature. It's where accounting meets agronomy and complex natural systems. and it can be time consuming. On this slide, you can see the nutrient budget for the practice spec for 590. Maybe your nutrient management scenario only applies to just one nutrient or one aspect of it. And I think that's where nutrient tools can help. With the right tools, and in this presentation I'm talking about online tools, you can streamline a lot of nutrient management. For one, you can just ask the questions that are necessary. You can also provide key information right when it's needed. If you need outside information to fill in a step in a nutrient budget, you can link to that resource. Tools can also increase efficiency in nutrient management. You don't have to do all these calculations by hand, and they can reduce the chance for human error. You can also strategically guide users to additional resources that are appropriate for their growing conditions, crops, and cropping systems. The first tool that I'll be talking about today is the Deep Soil Sampling Visualization app. This is an application I developed to help visualize deep soil sampling data coming out of the Lower Yakima Valley Groundwater Management Area, an area with high groundwater nitrate. The goal of this program is to test how nitrate varies with depth going down to 6 feet. On the right side of the screen, you can see some example data. This is straight out of Excel. It shows nitrate nitrogen, ammonium nitrogen, phosphorus, and potassium at various depths. For those who may not know what these numbers mean or may not have a good reference point, my question is, how can we make this data more accessible and more meaningful? So on the screen, you can see the Deep Soil Sampling Visualization app and the landing page for it. We'll start off on step 1. This is where you choose the crop that is growing in the field that you sampled from. We'll choose silage corn. As you can see, the rooting depth auto-filled with a default rooting depth. This changes by crop. If you chose wine grapes, that went to 5 feet, but we'll stick with silage corn. And then we'll insert a field ID here. And on the second page is soil test data. So the cool thing about this is you can copy and paste this directly from Excel, and I've already got this ready. But you can insert whatever values you have for whatever depths that you have. We'll accept the defaults for the remaining options. This page allows you to put a cost for your nitrogen fertilizers. You can estimate the value of nitrogen already in your root zone. We'll just choose those values here. And step 4 is where the visualizations appear. So for nitrogen, you have a soil profile, which shows these on a graph by depth. There's a crop need comparison, which shows the quantity of nitrogen and how it compares to the range of fertilizer recommendation ranges that exist in regional extension literature. There's a root zone analysis, which shows how much the quantity of pounds of nitrogen per acre in your root zone and below it, and then an economic analysis subtab, which quantifies that in dollar value. There's a tab for phosphorus and one for potassium. Those provide kind of guidance for those two nutrients, especially if they're in excess. On the next page, there's a downloads option, so you can download an HTML report that has everything you just saw in the previous tab. You can also download individual graphics. The last two tabs I'm excited about because these autofill based on the options that you chose. So some of these are the same no matter what crop you chose, whether that's, for instance, nutrient management, irrigation guides, those will be the same. But at the bottom, there are crop-specific crop guides that appear. And on the last page, there are links to technical service providers for further assistance. Our next app focuses on quantifying the plant available nitrogen released from manure. This nitrogen is released over multiple years, and how quickly this occurs and how much nitrogen is released depends on a complex set of variables. Here on the right, you can see a spreadsheet from publication EM 8954, a very helpful guide from Oregon State University that guides you through the various steps of this process. Our app, which you'll see on the next slide, aims to mirror that process in an interactive map that helps you rapidly compare different scenarios. So on the screen here, you can see the current iteration of the manure plant available nitrogen tool. You simply put your inputs on the left here. You choose your manure type, when it was applied, your application method, time to incorporation, and your application rate. These drop downs change their values based on what you inserted. So if you chose solid manure, you're going to see tons per acre and not gallons per acre. Down in the bottom left, you insert your manure test results. So that's going to be the percent nitrogen and percent ammonium. And we'll go ahead and enter a value for 2026. Now here on the right, you can see the release of nitrogen over time that we've calculated. The first year here in 2026 is the highest, and you can see how that changes over time. You can add previous years quite easily. Let's choose 2025. We'll add a slightly higher rate, maybe change the total nitrogen, and hit add year. And as you can see, the graph reloads, and you see the cumulative amount now in 2026, which is quite a bit higher. So you can rapidly go through a complex series of calculations that might take you quite a long time if you're doing it on paper, quite instantly, and see a helpful graph and a helpful table of those values. And lastly, I'll be talking about our nutrient management library that we built in Zotero. Zotero is a platform that is both online based, but you can also run locally in a program. And it allows you to easily organize libraries of publications, whether that's journal articles or in our case, land-grant university extension publications from across the Western United States. We've organized over 300 publications that are specific to crops grown in Washington State. And the great thing about Zotero is you can organize things by tag. So in the bottom left here on the screen, you just click on what crop you're interested in, and only those publications that apply to the crop that you're interested in will show up on the screen. You can also organize by year, so you can find the most recently published publications, or you can also sort by institution. All links in our Zotero library are checked weekly. They also serve as a single source of truth for other apps. So if there's a link in an application, it links to the same URL that's used in our Zotero library and that we're checking frequently. So it helps avoid any broken links. That concludes my lightning talk. Thanks so much for listening. If you have any questions, my email is on the screen. Please don't hesitate to send me an email. Thanks.
My name is Teal Potter. I'm a research associate at the Center for Sustaining Agriculture and Natural Resources. And today I'll be sharing some of the first 4 years of a 6-year study to assess whether we can split cover crop benefits between soil health and a biofuel feedstock. So this is an opportunity as Washington State and the West needs a lot more information on how to manage and grow cover crops well. And we also need to explore new ways to increase our biofuel feedstock, especially for heavy machinery and aviation that cannot be electrified in the near future. One question is, will we limit the benefits we could get from cover crops, in this case winter cover crops, if we remove the above ground biomass and sell it? Will there be any negative effects on the following cash crop by removing cover crops? On the other side, is this a potential way to get farmers to try cover crops if they could get paid? And for the biofuel producer side, this is an attractive, potentially stable source of biomass at an affordable price. It does seem to meet sustainability requirements. So in our experiment, we're growing cover crops at 3 different growing regions in Puyallup, Davenport, and Othello research stations. We are currently growing triticale and hairy vetch as our cover crops and rotating that with a cash crop that is winter wheat. And so here you see that we're removing the biomass from some of the plots and growing cash crops on all the plots following. And in Puyallup, we're growing sorghum, and in Othello, we're growing soybean hay. And at the Pacific Northwest National Laboratories, they are doing the economic and environmental assessment portion of the work for the potential to use this feedstock in a hydrothermal liquefaction pathway, which is a new but highly efficient conversion pathway for biofuels. So jumping right into results, just comparing the total yields of the cover crops at 2 sites here, our mix we have a mix of triticale and hairy vetch with 85% triticale, and the pure triticale stands perform much better than just the hairy vetch alone. We also are not seeing any effect of cover crop removal on the following cash crop at any sites. For soil assessment, we are not seeing any effect of removal on carbon or nitrogen pools or micronutrients, but I have the asterisks here to indicate that the first 3 years of this study, we were rotating plots. So we're not seeing the cumulative effect of removal. We'll hopefully get a little better data on that in the final 3 years. As for feedstock suitability, the composition of this biomass is fine for this HTL pathway. However, it would likely be used as a blend, perhaps with sludge, to be more economical and to have enough biomass to make it worthwhile. And for the economics, all the models were run at $60 per dry ton of biomass, which isn't nothing, but is probably too low to be attractive to most farmers. And for the environmental assessment, we do see that compared to fossil fuel production, we can produce a fuel from cover crops that requires 60% less emissions in the making of this fuel. So for more detail, we do have a couple articles out, a peer-reviewed article, as well as one in Washington State University's Connors News. So thank you.
Hi, my name is Gwendolyn Richards. I'm a graduate student at Cal Poly San Luis Obispo working with Dr. Charlotte DeCock. My project focuses on assessing the carbon storage potential of regeneratively managed vineyard soils, and this is specifically in California. So starting off, the overall project approach, it's funded by the Foundation for Food and Agriculture Research. We took over 200 samples from 12 different vineyards all across California, and the growers were extensively surveyed on what management practices they had been implementing. There was a wide range of practices assessed. Some were implemented for 5 years, some were implemented for more than 25 years. So our data set is a large range of different regeneratively managed vineyard soils. So we looked at specifically the implementation of cover crops, compost application, livestock integration, and no-till or reduced tillage. A lot of the vineyards did a stacking technique, so using, for example, cover crops with livestock integration or compost with no-till. So we're looking at the individual management practices along with what it looks like when we're stacking them. So more specifically, all of these regenerative practices increase the total carbon, but we're wanting to break it down even further. So there's particulate organic matter or particulate organic carbon and mineral associated organic matter or organic carbon. Particulate organic carbon is pretty quickly removed from the soil or quick to decompose, and mineral associated organic carbon is chemically bound to minerals in the soil and stays in the soil for a long period of time. So this is the kind of carbon that we're wanting to maximize. So far our results align with the saturation concept. So the thought is while the particular organic carbon can increase, this is the blue line, can increase pretty much indefinitely, the mineral associated organic carbon, the more stable pool, eventually the amount of minerals in the soil starts to level off or reach a threshold. And beyond that, there's no more long-term carbon storage possible for the soil. So what we're seeing so far is soils with more fine fractions, so with a higher percentage of clay and silt, they are able to store more carbon long term. And so what does this mean for growers? So a real world application would be obviously regenerative, sustainable farming is very resource intensive. And so the idea is that if growers could understand the potential and the limits of their soil to sequester or store carbon really long term, then they can allocate those resources for really the maximum amount of carbon storage. So there's been lots of research on carbon saturation as a concept, but the point of this project is to get region specific and crop specific so we can really apply the carbon saturation concept to better understand carbon dynamics specifically within vineyard soils in California. Thank you.
Hello, my name is Jadey Ryan. I'm a data scientist at the Washington State Department of Agriculture, and today I'm going to do a really brief overview and demonstration of Dirt Data Reports, a no-code soil health reporting tool. And this tool is developed for extension staff, technical service providers, conservation districts, and other soil health practitioners who have sampled from multiple farms and want to generate reports that are customized to each participating farmer. So a lot of soil health programs and projects collect soil samples from dozens or even hundreds of farms, and then want to provide useful information from those soil samples back to the farmers who have generously volunteered access to their fields and their time and knowledge. The workflow usually looks a little something like this, where you go out, you collect your soil samples from your field, you send them off to a lab, you get the results back in a spreadsheet, and then you have to generate all the tables and plots and maps and compile them into all these individual reports for each participant. Realistically, teams that are doing the soil health work don't have the capacity to manually generate reports in mass and bulk like this. Dirt Data Reports is meant to close this gap and make the reporting more efficient and more automated so that we have more time for outreach and technical assistance rather than manually compiling a bunch of reports. Let's get into the demo. So this is the Dirt Data Reports web app, and you'll be guided through 4 steps. The first is downloading the data template. So you choose if you want English or Spanish, and then click Download Template. And you can also click these hyperlinks for additional information. So there's instructions for how to fill out the template. So the template will come up in your Downloads folder, and then you can open it. And there'll be 3 tabs: Instructions, Data, which you'll replace this example data with your own, and a data dictionary that tells the app how you want your measurements to appear as far as their names, their abbreviations, and units. So after you follow the instructions and replace the example data with your own, you're ready to move on to step 2, uploading your data. And you'll upload, for the demonstration, we're just going to upload the template that we just downloaded in step 1. And it goes through a bunch of data validation checks. And if they all pass, you can move on to step 3. If they don't pass, then the errors that you need to correct will be listed here in a red box. So moving on to step 3, this is where you customize the reports with your project-specific information. So the project name, summary, which measurements you have had analyzed. And once you've filled out these sections, the text, you can preview the report and see which measurements you have chosen and how the text appears in each of these sections. And once that preview looks good to you and you're done editing, you can go to step 4, where you will generate your report. So right now, you can only select up to 5 producers to generate the reports at a time. We're working on increasing the speed of the report generation so that we can do more than 5 at a time. But for now, 5's the limit. You choose if you want Microsoft Word or HTML reports, and then click the Build button. And then this pop-up will tell you which report it's working on. And this can take a few minutes, so don't exit the session, which means don't close out of this tab or this browser unless you want to restart this process. So once the reports have been generated, you'll get a little pop-up, and a zip file will appear in your downloads. So we now have a zip file, soil reports, that has our Word document and we can open it, and we have our report. And that's a really, really brief demonstration. So I encourage you to check out the app yourself and try it out, even if it's just running the report with the example data. There's a blog post for more information, open source code, and we also have the Soils R package from which Dirt Data Reports is built on if you have the coding capacity and you need more advanced customization. Thanks so much for tuning into this talk.
Hi everyone, my name is Tejinder Singh and I'm a PhD candidate at Utah State University working with Matt Yost. Today I'm going to talk about how continuous field measurements can shift water retention from a static laboratory property to a dynamic soil health indicator. Soil health assessments are usually about how soils function, and one of the most critical function is how our soil stores and supply water to the plants. This helps a lot in a crop stress controlling, productivity, drought resilience, and many other factors. Despite how important this soil water retention is, it's rarely measured under real field conditions. Most of the assessments are usually done in a laboratory conditions and do not capture how soils behave under real field condition during or within or throughout the growing season. Let's start with what exactly soil water retention is. Soil water retention is how water is held in the soil. At saturation, all the soil pores are filled with water, but due to the gravitational pulls, water drains rapidly and it is largely unavailable to the plant. But as the gravitational water drains out, our soil gets to the field capacity. That is point where water can be extracted by the plant. So it's stored in the meso, macro, micro, all the different kind of pores during the field capacity. As we drains more water or plant extract more water, we get to the permanent wilting point. That is the limit or the lower end where no more water is available for the plant. And the range between the field capacity and the permanent wilting point is called plant available water. That is actually how much water a plant can use. Since soil health practices aim to improve structure, aggregation, and pore connectivity, these changes are usually expressed through the changes in the soil water retention behavior. Soil water retention is usually measured using all the different types of laboratory methods and all the different kind of instrument, but there are key limitations like these are destructive laboratory based soil cores we have to remove from the field. These are static measurement single estimate for an entire growing season, and they cannot detect the short term soil health changes we are seeing after our practices. So this really raises a question like, can we really measure soil water retention directly in the field under natural field conditions? And the answer is yes, we can. So what we are exactly doing, we are trying to install co-located sensors, including volumetric water content and metric potential sensors together at the same depth and collecting data during the same interval of time. Once we install the sensors and get the data, we are extracting the drying period between two irrigations or between the precipitation we are receiving. Once we get the drying period, we are deriving in situ soil water retention curves from the field data, real field data. As soon as we get the retention curves, we fit in using different models to see which exactly model works best and see can these works best for the field installed sensors. And then we're going to be using it for further predicting all the different properties we want to see. So what we can actually quantify from this in situ installation, we can get the soil water retention parameters from which we can calculate the unsaturated hydraulic conductivity of the soil. And we can also get the water retention indicators like field capacity, permanent wilting point, plant available water, which are really critical for the crop to be understand. Why this method is ideal for soil health assessment, why we are focusing too much on like co-location. So whenever we apply any amendment or we follow any soil health practice, we usually apply in the topsoil any kind of amendment. And that's where we usually want to see the changes in structure and aggregation. After years of research on the co-located sensors, we found out the better retention curves are usually obtained at the shallow depth. So because we get the longer drying period between the rainfall or irrigation at the shallow depth. So we got the right place where exactly it works best, and we got the right method which also works best at that particular zone. And if we combine both of them, so we can get the strongest soil health signal in our field under real field conditions. Why this method matters for the soil health and resilience: it will capture subtle, small changes in the structure and water retention within and across the season beyond the yield responses. It will track how soil health practices and weather condition uh, function together within the season, and it also connect how soil water buffering to drought impact on crops, food system, and human health. Thank you.